Hi everyone. I'm really happy to welcome you to this lecture on transcriptomics and Venomics and broader applications of that in the training school from the European Venom Network. And I have to apologize in the beginning. I was really planning to give this lecture live, which I always prefer, also question-wise. But I'm currently on full parental leave and doing, let's say, things more than 45 minutes on block is at the moment a bit difficult. So our little daughter is lovely, but this is the caveat with this. You will see me drinking a lot of tea and coffee during the lectures. But I wanted to at least introduce myself a bit, well, let's say as live as possible. So I would say we start, and maybe this is now the time to grab some tea or coffee, or maybe beer or wine, depending on you are, or a nice juice. And yeah, let's start. All right. I thought actually in the very beginning I would like to say some words about myself. So who am I? Teaching you today, so I have a better idea of what my background is. I would say I'm not the typical venom researcher. I come from zoology and evolutionary biology, so I'm more linked to the biology of species. My expertise is in particular molecular, so I work on molecular systematics and phylogeny, using also phylogenomic data to analyze relationships of species. I worked also on phylogeography and speciation. I'm still interested in that field. And my core taxon expertise is arthropod evolution, in particular higher arthropods and pancrustaceans. So the pancrustaceans are insects and crustaceans. And I could show with first phylogenomic data that insects evolve from a crustacean group, which are the remipedes, which reasons also the name Pancrustacea. The remipedes were also then finally that taxon which brought me to venom research, because remipedes, which you can see here, were supposed to be venomous, and no one really looked into that in more detail, which I did then together with Ronald Jenner at the Natural History Museum in London, and we could show that indeed remipedes are venomous and could add a first venom description for crustaceans to all the other venomous taxa. This slide shows you basically what's about for me. It's about the species, so I really like to be in the field. That's the reason why I started this actually a long time ago. And I have not so much a preference. I'm interested in many species, also the non-venomous here are shown the venomous though, except maybe the Tsuganids, which are poisonous. But I like also to be in the field, no matter if it's aquatic or terrestrial. I have kind of a favor for aquatic organism, despite that isn't reflected so much in my venom research yet. And I focus taxon-wise on remipedes. I'm still working on that and more specific toxins, but I also have a strong interest in robberfly venom evolution or dipteran venom evolution in general. And recently I could obtain also funding for hymenopteran venom evolution, where I work particular on higher hymenopterans, aculeates, solitary bees, and also the true wasps. There are some other studies, for example, on marine ribbon worms, which I could do. And in the last years, linked to the LEVE-TBG consortium, I tried to add also the genomic perspective on my research. And about this you will hear in the very end of this lecture, to see why that is actually important, in particular if I want to talk about venom genes. There's a difference between venom proteins and venom genes, yes. My current role in the European Venom Network is that I co-proposed this cost action and currently lead Working Group 2. In Working Group 2, we try to describe and push a bit further the best practices and innovative tools in venom research. So it's a group of very mixed background of involved people, and we could recently publish one of the first white papers of the European Venom Network, which you might have seen already, So maybe check it if it's interesting. If you have maybe ideas, or if you don't agree in some areas, then please join working group 2. We want you, and maybe your similar ideas, or maybe also particular the other ideas, and then we can push together the best practices, innovative tools in Venom research. So this lecture, just to give you an overview or warning ahead, I'm not sure, is substructured, and each of the topics where you see the headings here is kind of supposed to be an own block. So you could generally watch or listen to that independently from the others. I'll give a very brief introduction, not to overlap too much with my other colleagues in the other talks and lectures. Then we talk about pre-planning and RNASeq projects, which I realized is very often neglected. The sample strategy, specimen species-wise. And then we talk briefly about the sequencing platforms, including also long read, not only short read sequencing. There are 2 additional blocks that are included here now, which don't hurt. So basically all or most of the RNA-seq techniques come from DNA sequencing and genome sequencing. And so to know something about the history of DNA structure, but also sequencing cannot really hurt. Then we talk about the sequencing process, and then the RNA extraction and library preparation, with some points important for smaller neglected venomous animals. The read assembly, and then after we have assembled all those reads into transcripts, what they transcripts could be, which is a transcriptome annotation, basically predicting genes, gene model prediction. And then briefly about the quantification, some words. Particularly those 2 topics here could be an own lecture. So we'll focus a bit on the general cases in Venomics. And in the very end, I would like to give a genomics perspective, also trying to make that short, because now we will have soon so many genomes available that you hopefully see after this lecture why that is important, what these techniques add actually to the Venomics research. All right. All right, as you probably have heard already, Venom research or Venomics is a quite diverse field, and it's linked to many different aspects, like translational aspects using toxins for applied research. And I will focus in this lecture a bit on that part here, where we have transcriptomics, proteomics, morphology, and genomics as the core pillars of venom research to describe venoms of animals. And I would like to highlight one point here, which is a technological perspective. So you see here those machines, mass spec and sequencer. This is extremely important right now. We have an incredible speed of sequencing technologies that are opening new doors. And I'm not sure if I can still keep track right now. I'll try to give you an overview of the most recent technologies here, also direct RNA sequencing, for example. So keep that in mind, that the technological perspective is very important, but also the species perspective. So those 4 main research fields, transcriptomics, proteomics, genomics, and morphology, describe, of course, venom composition of species which are interacting with other species, maybe shaped by these interactions, but also are impacted on extrinsic factors from the outside, like human growth, climate change, etc. And these are maybe all processes that we can tackle, bringing those 4 research fields together. So today is about the 3 major omics, which is how I like to call them. So the transcriptomics part you can see here in the middle. This is a very brief overview of processes to finally describe a venom profile of a species. I like to highlight here already in the beginning that this lecture is not about RNASeq or transcriptomics alone. It's not like transcriptomics is the holy grail for venom research. No, of course not. it's important to link that with the proteomics and also later to the genomics. So I'll try to bring that all a bit together, but focused on transcriptome analysis, because from my experience, this is the part where many people lack a bit of expertise, especially because they come from different fields. So no worries here. And the goal of, or the aim of this lecture is that I would like you to know how to analyze a Venom profile for species using prototranscriptomics, which options you have, and why and how you can use those, and how to start basically planning the analysis or project for you. The genomics perspective, I mentioned it already. I like to add here that you understand why complementary genomics is important and why and where it should be used. And in the end, you should be aware of the diverse aspects on Venom protein annotation and Venom genes. You should know how to avoid annotation errors, how you maybe not present expression level data or figures, and also that you refer to genes if you talk about genome data actually. And yeah, I hope I could manage to summarize that in a nice way. I hope you realize during or after the lecture that this is actually a topic for an own seminar. So I really did my very best. All right, very important point before you start the actual RNA-seq experiment is the pre-planning, which you can see here. We talk about this flow here in this block, which is the specimen collection, which normally happens in the field. Then the laboratory part, where you maybe manipulate your species. So if you're interested in predatory and defensive venoms, for example, you tickle your whatever species a bit differently to provoke defensive and predatory venom ejection. And then comes the part here, which is a favorite one of mine, that you actually have to describe maybe the venom system or the venom apparatus of a specimen ahead experiments you do, because maybe if you reconstruct it, you will find some surprising results that can change your perspective on the species and gives you new ideas. So let's start with the field. My background is very much linked to natural history museums, so this part is quite important for me, and I was also trained in that. If you target a species to describe their venom, an important question if you start planning the project is that species maybe protected? Do you need a permission to collect it in the area? Is it a private area, or is it maybe a regional area that's protected, like a nature conservation area? Is the species itself maybe protected on a local or countrywide protection list or red list, like we have in Germany? And then a very important part is the country you collect the species in, part of the Nagoya Protocol. Is the research you do basic research, like evolution of venom proteins or the general description of the venom composition, or is it maybe applied research where you want to use an identified toxin to develop, I don't know, the new blockbuster pharmaceutical? Both cases are treated differently regarding to the Nagoya Protocol. So you do well if you check the site, which is the Access and Benefit Sharing Clearinghouse of the Nagoya Protocol parties, which you can see here. And there you see which country is party of that Nagoya Protocol, and you also see the contact of the focal point office that give you all further information, what you have to do and how you have to do it to collect with legal permissions the species you aim to study. Then comes the part where, depending on the research objectives and species size or abundance, you have to think about is a species suitable for laboratory capture or manipulations after the capture. So normally it makes sense to synchronize the venom glands of individuals if you want to pool them or describe the venom composition. So you have same conditions. So all venom glands are kind of replenished at the same timeline. And then you can also assess, for example, different temporal venom expressions. So you can say, okay, the specimen ejected the venom, and then you can see how in the different days or hours, depending on your species, the venom profile changes. And of course, you can then also make better estimates of defensive and predator venom composition. But this stands and falls with the question if your species are suitable for laboratory manipulations. So many species I work on were a bit surprising on that aspect. So I thought Hymenopterans super cool, easy to milk. And especially with solitary bees, it becomes really difficult. So test that and think about that. And then comes also the question linked to the kind of study you want to do. If you want to describe generally the venom composition, or if you want to go beyond that, you need maybe biological replicates. So if you want to do an expression profile, for example, then you need to ensure that enough replicates are included to have a clean statistics. So you need at least triplicates or more. If you talk to ecologists, at least 5 or 7 replicates to ensure that you actually talk about significant changes. So this is needed for differential gene expression studies, which is, from my perspective, really tricky for me because I work with smaller individuals. So that are all aspects I learned partly also on the hard way that this is something you need to think about before you start the experiment. And then finally, what is the venom system? Do you know it already? Is it known, or is it smaller or larger, or do you have a distinct venom gland? So you have basically lumen in which venom proteins are secreted, or do you have, we talk about that later, like distinct glandular cells which secrete venom proteins? And those are really tricky ones. So I'd just like to show you that you can really do pretty cool stuff with actual morphological methods. So I used normally and still use for my species, I study synchrotron microcomputer tomography. So basically it's like a desktop microcomputer tomography. You just go into a facility that has a synchrotron, which is a particle accelerator ring. And then you can take your samples there and do images of those, which are stacked images, and then you can do final 3D reconstructions. So for desktop micro CTs and synchrotrons, the preservation of individuals or samples is pretty much the same. I always collect them in Buin liquid, critical point dry, and then mount those samples, which you can use then for micro CT or synchrotron CT. And if you're very careful, you can also reuse them. So basically, just very fragile dry hulls. And then you do those segmented stacks. Partly you have to still manually segment those pictures or single images, and then you can do a cool 3D reconstruction that would give you maybe new insights of the venom system. So this is now the venom system of a robber fly, where you can see if you eliminate all the chitin structure, that those have actually 2 venom gland systems. So in blue you see here the thoracic glands, then in green the duct, and here in red there's this proboscis gland system, where I was not sure if it's actually involved in the venom secretion. We could show with our data doing differential transcriptomics here that we do not find evidence for expressed and secreted venom proteins in this gland. However, it's also still a bit preliminary because with proteomics techniques, these are very difficult structures because they're so small. So this would be now maybe something for the new multi-imaging techniques to check that for sure. But keep that in mind. If you tackle an interesting species, maybe there is something hidden in the Venom system or Venom apparatus. You don't know that. So it makes maybe sense to do ahead of your project strategy a scan of the species to see what's actually going on with the Venom system. So basically those are the 3 points that are important for the pre-planning before we go into the sample strategy of the RNASeq analysis. So let's talk about the sample strategy and different cases of more or less easy venomous animals to study. So basically we talked about this flow here now, and now we talk about this part where we go into transcriptomics. And well, let's say you have an insect like a robber fly and you want to describe an unknown venom composition, then you would normally link proteomics and transcriptomics immediately, where you collect the crude venom of the robber fly, which is basically that venom that is being injected into a possible prey or against a predator to defend itself. And then you would use the venom gland tissue to compile a transcriptome of this venom gland, which are the expressed precursors of the venom protein. And let's say in this case, it's very easy to milk those individuals, and then you can basically just milk them and then dissect them. And he already highlighted, if there is a genome available, please mine and use that genome also. We'll talk about this later. So right now we have an incredible number of genomes popping up from all these genome consortia like ERGA or the Sanger Sequencing Consortium in the UK, European Genome Reference projects, etc. And it can well be that you actually work on a species where the genome is already accessible, and this will have many advantages. But so this is now a good case where we can basically use the proteome data as a baseline for the transcriptomics. So we obtain the venom, then this is used for mass spectrometry. That's the talk by Sebastian. And we compile a transcriptome or sequence a transcriptome of the venom gland tissue that is then basically used as a database for the mass spec analysis. So we have this hypothesis that this is a new species and maybe also the venom proteins are new, so we need a tissue-specific database to identify the fragments from the mass spec analysis, which come then from the transcriptome. Very important here. This is basically the crucial link between proteomics and transcriptomics, making it proteotranscriptomics. Question here, why is the proteome data so important? Maybe think about that a few seconds. Yeah, I've mentioned that already a bit. It's the best way to address the venom proteins only. Because in the tissue of the venom gland, we have, of course, not only the precursors of the venom proteins, but also the other gland proteins, which are basically housekeeping genes to keep the gland running and functional. So the venom gland tissue always contains non-venom proteins. Many of them are belonging possibly to the same family, like some of the venom proteins, so you have false positives in your prediction later. So you use, if possible, the injected venom proteins at the threshold, and then you discuss in your proteotranscriptomic study basically only those that match the proteome data, and the other ones you ignore. Of course, you still show them in the supplementary file of the transcriptome annotation, but you better just discuss the ones that match the proteome. And here the question, what is actually the challenge with transcriptome-only studies? I think you can answer that now quite easily. It is indeed the identification of multi-gene family variants of venom proteins is limited and remains very putative. So if you don't have the proteome as a threshold, you might list variants of gene families which are actually not in the venom gland, but keep the venom gland running as housekeeping genes. So this is a very important part. And hold that thought. We come back to this later. But yeah, so this is basically a good case. We have also cases where it's a bit different. Maybe just as an additional slide here, often pooling for RNA-seq is necessary. So you have to pool multiple individuals to gain enough tissue to sequence venom gland transcriptomes, especially if you work on smaller species. The advantage here is that the transcript populations are more exhaustively covered, so you maybe describe the venom composition a bit better for that species, not for the individual or specimen. But the disadvantage is that the transcript diversity increases, of course, linked to individual variation. So you can overestimate actually the diversity of the venom proteins you identify using pooled samples and the transcriptome. Please keep that in mind. And then we have a case which I know quite well, where the milking or stimulation is not really working out. Referring to solitary bees, they are really happy to sting you, but to give the venom in a kind of a standardized manner into a tube is not that easy. And then you have to dissect the venom gland kind of simultaneously. You basically microdissect the gland, and then you obtain the protein liquid from the gland by squeezing it out. So if you work with insects, they normally have a very robust chitinase structure and body wall, and then you can squeeze it out very nicely, and you can see this Vaseline-like liquid coming out into the millipore water, where of course you need to be aware that you will have some contamination with other gland proteins that are not linked to the venom gland or just by the dissection. And the same here, of course, this doesn't change. The tissue for the transcriptome contains expressed venom proteins and other gland proteins. So the solution here is to include also body tissue, if possible multiple body tissues or a complete body. So you have basically all gene variants and proteins covered, and complete gene completeness ensured. And then you can differentiate between maybe gland unique proteins you identify in the transcriptome that only occur in the venom gland, while you have then also variants in the body tissue which maybe occur in the venom gland, and then you can differentiate a bit better, and you can maybe sort out some interesting and unique proteins in the venom gland based on this comparison between body tissue and venom gland. In general, in that case, it's important to discuss the data conservatively. And then we have also the ugly cases where we actually do not really have a distinct venom gland, where it's really a bit difficult to milk it properly. So this is a larger weaverfish I work on together with colleagues at the Liver TBG consortium. So we have sequenced the genome, but those fishes have linked to their spine not a distinct gland system, but tissue with gland cells. So dissecting the gland is very difficult, and the milking is the only way to do it in the best way, but you will have also contaminations by skin, etc. So this is not so easy to do. And the way out here is also to use maybe a comparative expression profile of spine tissue and other tissues, plus the milk venom. Linked to the genome, we have of course multiple tissues taken, so gene completeness is ensured there. And this is one way to do that. Where it's a bit more difficult in an ugly case is, for example, with ribbon worms. They are also difficult because they have separate venom gland cells, and they're difficult to milk. We could describe here the first prototranscriptome data on one of the hopefully immersion species. They have kind of a tooth structure at their proboscis, so they really can pierce other worms. And there we used basically body slime, mucus, and proboscis proteome samples and compared them a bit, but we could only do a proboscis transcriptome. So our data was quite preliminary. So I was really happy to see the work on a similar ugly case by Eda Verdis and colleagues, who did that in the best way you can do it, actually, where differential expression profiles of the proboscis tissue and other body tissues were done. So we have replicates and can really say something about differential expressed transcripts. And then multi-imaging was done, where you can see also the location of the expressed transcripts in the proboscis, where you can then identify putative toxins. So this was basically giving you an overview of different cases for venomous animals you may like to study, and think about that very carefully, how you design your experimenter, but especially also how you interpret then in the end the data. Please note that RNASeq has its limitation, and the combination with proteotranscriptomics is really important here, and later we see why genomics can also help there. All right. You probably think now, great, I have now my project planned. Which sequencing platform should I use now? Sequencing. Well, there's Illumina, as you probably know, then there's PacBio, and also ONT, OMG. So we have three different ones. We have short reads, we have long reads. I'll try to give you a brief overview to help or assist maybe with the decision where which platform makes sense. Basically, we talk about this part here now, the transcriptome sequencing. So most of you probably know the Illumina-based short read platforms, where you extract RNA, do lip prep, sequence complementary DNA, and then you go into the sequencing. And then you have a lot of short reads, and you process them. You assemble them by breaking them up in shorter reads, which sounds counterproductive, but actually helps. And then you have the transcriptome, which you annotate, and then follow all the subsequent analysis you plan. And now we have also long read sequencing technologies, which are sometimes referred to as third generation sequencing technologies, PacBio and Oxford Nanopore. There are some differences compared to the short read sequencing. So we have an RNA extraction here, and cDNA library preparation and complementary DNA generation for those two systems. But we have then a different clustering, so it's not really a k-mer assembly like with the short reads, but it's kind of a composed assembly. We talk about that a bit later. But then we have also from Oxford Nanopore Technologies the possibility to sequence RNA directly. So basically here we don't prep any library, so we have no library errors in there, but we anneal primers and then we sequence the RNA. So these are basically the three options you have. You have short read, long read, and direct sequencing, which is also a long read sequencing. And in general, you can try to make a decision using a classic tool from project management, which is the golden triangle. It has nothing to do with drugs, it's actually a decision helper. So we have a cheap price for Illumina sequences, and PacBio and ONT are still a bit more expensive. It depends, of course, on the conditions you have. Maybe your facility or institution has special deals, or even the machines in the laboratory, or a working group has, for example, ONT min-ion established, then it maybe makes sense to go for that. And it's in the end not that expensive. Important, however, is the input you need for the sequencing of the RNA. So for Illumina, you still need the lowest input possible. So this enables you to sequence really very small samples, while for PacBio and ONT you need more. We talk about this a bit later. We talk about the RNA extraction. And then we have, of course, the quality topic. So for Illumina, you still have some assembly artifacts because you work with short reads. And you have a high coverage. So we have, if you use Illumina, you have really a lot of reads with a high coverage. While for PacBio and ONT, we have a lower coverage because you sequence longer molecules. So the number of reads you obtain is lower. Here we have an amplification step, and here we have no amplification. Keep in mind that ONT and PacBio also are suitable for methylation patterns. So that is maybe important if you want to sequence a DNA. And then comes the part that's important for RNA. Complex isoforms detection, of course, is much better with the long read sequencing than compared to Illumina, which is linked to the assembly. And basically with the long reads you sequence the whole mRNA, so the whole transcripts that are expressed, while with Illumina you sequence fragments and then you need to reconstruct those transcripts. So isoform detection, if you want to do that, then you have to use, of course, the long read techniques. This field is in the moment quite in motion. So from what I know, also from colleagues, you normally sequence both. So for long read sequencing technologies, you normally add always Illumina to polish possible sequencing errors. But that might have changed now. I didn't find anything though. So keep that in mind. So I think most of you will go for Illumina, which is still the most established sequencing platform. Also the errors that are created by this platform, like assembly artifacts, chimeric transcripts, etc. They are quite well known, and there are always workarounds, more or less, for those cases. While some of the long read techniques and also the kits are still a bit in progress. Before we go into more detail now regarding the RNA extraction and the cDNA library preparation, after this brief overview, I would like to squeeze in actually those two blocks on history of DNA structure and DNA sequencing, which help, I think, to better understand the processes we talk about later. And I think in general that knowing a bit about the DNA structure helps at all, in general. And in particular, in this part, there is a itsy-bitsy lecture on good scientific practice and women in STEM involved. So maybe check this out. While working on DNA and RNA for longer now, I'm always still astonished that we didn't know for very long which molecules actually inherit traits. So this journey started 1868 when Friedrich Miescher described at that point an unknown compound from nucleus cells, which he called nuclein, which we know today were nucleic acids. And an intensive discussion began then in the 20th century. What is the substance of inheritance? Main suspects were actually proteins for a longer time. Until then, in 1944, Oswald Thierry finished experiments from colleagues and showed that DNA is a substance that enables the transformation of harmless into deadly bacteria. And then the question was a bit the structure of the DNA. So you've probably heard of Aaron Chargaff. We still know his two rules. The first one described basically that the bases have relations. So adenine is always linked to thymine and guanine to cytosol. And this is something we still know, that we have those pairings. And he showed actually also, which is the second Chargaff rule, that the DNA composition varies in species. So some have a higher GC content. And then comes the question, you probably know who discovered the structure of DNA, or do you? Most people link James Watson and Francis Crick to the structure discovery of the DNA. They published a model of the DNA structure in 1953. Interesting is that the first model they actually assumed was incorrect because they ignored, like, magnesium has a different condition in natural systems. So you can see here, this is a bit odd as a structure. And only photographies of Rosalind Franklin, researcher who worked on crystallography and a lot of other fields, actually showed the correct helix structure, where she also discovered the different forms of the DNA. So everyone who extracts DNA knows that there are different conditions of DNA. This is the picture of A DNA, and this is the famous picture 51 from the B DNA structure that Rosalind Franklin did. So the B form is the one that actually occurs in standard conditions in biological systems. And if you add heat or stress in a large quantity, then it goes into the Z form. If you reduce water, then it goes into the A form. The Z form is that one that inhibits actually any sequencing or PCR amplification if you left your DNA on the columns after extraction too long in the heat cupboard. And the role of Rosalind Franklin was very long overlooked, and I always take this opportunity to show this story, actually, talking about DNA. Her name definitely should be mentioned, and for that reason I included a short video here that I would like to share with you.
The discovery of the structure of DNA was one of the most important scientific achievements in the last century, in human history, in fact. The now famous double helix is almost synonymous with Watson and Crick, two of the scientists who won the Nobel Prize for figuring it out. But there's another name you may know too, Rosalind Franklin. You may have heard that her data supported Watson and Crick's brilliant idea, or that she was a plain-dressing, belligerent scientist, which is how Watson actually described her in The Double Helix. But thanks to Franklin's biographers, who investigated her life and interviewed many people close to her, we now know that that account is far from true, and her scientific contributions have been vastly underplayed. Let's hear the real story. Rosalind Elsie Franklin was born in London in 1920. She wanted to be a scientist ever since she was a teenager, which wasn't a common or easy career path for girls at that time. But she excelled at science anyway. She won a scholarship to Cambridge to study chemistry, where she earned her PhD, and she later conducted research on the structure of coal that led to better gas masks for the British during World War II. In 1951, she joined King's College to use X-ray techniques to study the structure of DNA, then one of the hottest topics in science. Franklin upgraded the X-ray lab and got to work, shining high-energy X-rays on tiny, wet crystals of DNA. But the academic culture at the time wasn't very friendly to women, and Franklin was isolated from her colleagues. She clashed with Maurice Wilkins, a lab mate who assumed Franklin had been hired as his assistant. But Franklin kept working, and in 1952, she obtained Photo 51, the most famous X-ray image of DNA. Just getting the image took 100 hours. The calculations necessary to analyze it would take a year. Meanwhile, the American biologist James Watson and the British physicist Francis Crick were also working on finding DNA's structure. Without Franklin's knowledge, Wilkins took Photo 51 and showed it to Watson and Crick. Instead of calculating the exact position of every atom, they did a quick analysis of Franklin's data and used that to build a few potential structures. Eventually, they arrived at the right one. DNA is made of two helicoidal strands, one opposite the other, with bases at the center like rungs of a ladder. Watson and Crick published their model in April 1953. Meanwhile, Franklin had finished her calculations, come to the same conclusion, and submitted her own manuscript. The journal published the manuscripts together, but put Franklin's last, making it look like her experiments just confirmed Watson and Crick's breakthrough, instead of inspiring it. But Franklin had already stopped working on DNA and died of cancer in 1958, never knowing that Watson and Crick had seen her photographs. Watson, Crick, and Wilkins won the Nobel Prize in 1962 for their work on DNA. It's often said that Franklin would have been recognized by a Nobel Prize if only they could be awarded posthumously. And in fact, it's possible she could have won twice. Her work on the structure of viruses led to a Nobel for a colleague in 1982. It's time to tell the story of a brave woman who fought sexism in science and whose work revolutionized medicine, biology, and agriculture. It's time to honor Rosalind L.C. Franklin, the unsung mother of the double helix.
Right, I like to add that I use this lecture and also this video always as an opportunity for myself to kind of readjust my good scientific practice. I think it happens unintentionally that we forget people or do not acknowledge people who contributed to research projects, and this is actually important to continuously kind of check that we don't forget that. It's really important. Before we look a bit more detailed into the sequencing process, I'd like to give a brief history of the DNA sequencing, kind of a journey through the history of DNA sequencing, which I actually picked up from Twitter, this photo. Here we have basically the starting point with the discovery of nucleic acids, 1869, without knowing that's actually DNA. And then we have 1953 with the publication of the double helix structure of the DNA. And 1977, a very good year, the first generation sequencing was established by Sanger sequencing, which became then more feasible and practical after the invention of the PCR, which actually happened later, I think 1985 or 86. And then it took a while until 2005, next generation sequencing was invented, and the reign was opened by the 454 pyro sequencing platform from Roche, soon followed by the solid sequencing by ABI, and then finally we have 2008, the Illumina platform in action. The third generation sequencers are also referred to as long read sequencing technologies, and PacBio is here working in 2011 with his first generations of smart cells, and in 2012 the first generations of Oxford nanopore technologies were usable. So this is just a brief overview and shows you how fast those technologies are developed, and that there will be a lot still to come. All right. Sequencing processes. You're probably surprised that I didn't use all right. I like to start here with an old school player, which is Sanger sequencing, invented by Frederick Sanger and Alan Coulson. You see those two colleagues here, which is also called chain determination. It has a very low error rate still, and produces, well, these times not that longer fragments anymore, but it's still quite in use, especially if you do population studies using single copy genes or DNA barcodes. Then sometimes Sanger sequencing is still applied. Important is here that we have a separation of two reactions. The first is a chemical reaction, the second a physical separation. The chemical reaction is also called cycle sequencing. That's basically a bit like a PCR, just that it's linear. So we have here primers, we have a polymerases, and we have the nucleotide triphosphates, you also know from the PCR, but we have a second kind of nucleotide triphosphates, which are the dideoxynucleotide 3-phosphates. And they differ in the way that the sugar is modified here, and there can no other nucleotides be added anymore, and they're dye-labeled, so there's a fluorescence marker added to the base. Well, let's start this reaction here with my great old-school animation. So we add the primer to the sequence, and then the polymerases also attaches to this, having a starting point, and then the nucleotides are included and the chain is extended. And it's extended until one of the DDNTPs is integrated. Then this reaction here can't proceed anymore. The chain is determined. And then the next nucleotides are incorporated until the next DDNTP is incorporated. And in the end, you have a lot of different double strands of different lengths. And this happens all in a PCR, like a tube, or later then also on a microtiter plate. And this here goes then into an electrophoresis, which is a second step. So you have a separation on polyacryl gel matrix, and the separation occurs then according to the lengths. And then the bases are called, which means that the laser stimulates those dyes attached to the nucleotides from the DDNTPs. And this is then basically the base calling, what happens in the computer, and those are the electropherograms that students like so much. Clipping and trimming primers, understanding also how forward and reverse primers work. So you have those sequencing editors like Bioedit and Genius where you can do this. And this is actually the sequencing technology where you still see those electropherograms. I used actually IBI capillary sequences and the famous 377 plate sequencer in my diploma thesis and later then in my PhD. So I did the sequencing myself, which is now with the next generation sequencing not really possible anymore because it's normally outsourced to the companies. Which brings me now to the pyrosequencing invented by Paul Nuren and Mustafa Ronagi, which was the first high throughput sequencing technology, especially linked to the platform developed by Roche. It's also called real-time light detection principle, allowing for massive read numbers because the whole process could be parallelized and automated. What we have there is also the same principle with primer polymerases and then of course the sequences, but we have also sulfurases and luciferases, which are important for later light detection. So the primer adds here a starting point, then you have the polymerases that is added, and then you have separately for each nucleotide kind of a washing system where the nucleotides are poured into the system, and this time it's ATPs. It's not added because we need here we have a cytosine here, and then we add finally here the GTPs, and then by the incorporation pyrophosphate is detached from the ATP, and with the sulfurases an additional adenosine phosphate sulfate and ATP and the luciferases, we have a light reaction because luciferase is one of those enzymes that stimulates in the end a light emission. So this is kind of around corners, but the cool thing is you can parallelize that. And particular important here is also the invention of emulsion PCR, which means that the sample processing and sequencing is automized. So for pyrosequencing, you normally use a genomic DNA or RNA, and then you fragment that physically or chemically. This is basically the same process that we also use in Illumina. So actually I did that linked to my PhD where I used pyrosequencing facilities at the Max Planck Institute, and we did the sample preparation. And then you ligate adapters, two different ones, A and B, and then you receive in the end single strands with both adapters attached. And those single strands you can then bind to a bead. That's basically a chromatic compound where the one of the adapters adapts to a bead adapter, and then you can bring this with the PCR chemicals and emulsion oil into a microreactor. Here's a tube, but actually it happens then on a picoteter plate. And then one bead each sinks down into a well of this picoteter plate, which you can see here, which is then brought into the sequencing machine, where the reaction I just explained happens then. And there, for each base separately, you have those chemical tanks, and then the system is always flowed with each base separately. They are cooperated, light is detected or not if it's not incorporated, and by this automated process you end up in a pyrogram where by the light reaction you can calculate which nucleotide was incorporated. So you do not have anymore a direct electropherogram like with Sanger sequencing, but first computationally based a pyrogram where you don't see the direct sequence anymore. This is no longer used so much after Illumina sequencing, sequencing by synthesis was invented by those two colleagues here. And here are two principles important: sequencing by synthesis and the flow cell bridge amplification. So that means amplification and sequencing occurs at the same time in real time. So the principle here is also that we have a primer and a polymerases, but we go back to fluorescent nucleotides that have distinct colors and that are blocked chemically. So the primer attaches the polymerases as well, and then the nucleotides are added, incorporated. They do a fluorescent reaction, and then nothing can happen because they are blocked. So they're deblocked in a washing system, and then the next one is incorporated. Fluorescence reaction occurs. You can see, okay, it's a T. It's deblocked, and this goes on the whole time until we have the sequence. And then follows the same principle we know already from the pyrosequencing. If you have the genomic DNA or RNA, you fragment those longer double strands into smaller ones, either by ultrasound or physical or chemical by enzymes, and then you add here also adapters. And those adapters attach then to those flow cells here, which have also adapters attached to the flow cell. And then you have those single strands which move on this flow cell, and then they build a bridge, because the other adapter can attach to a counterpart on the flow cell, which is the bridge that is built. So we have free termini here, and then they can be kind of detached. And as soon as they're double-stranded after the reaction occurred, we have kind of those clusters that build up, which is basically amplification. And then, if now the bases are added, as I explained before, then you basically have the sequencing by synthesis. And I would like to show you for that a video which makes that maybe a bit more feasible to understand. I work always with moved pictures better than with the explanation like I just did. So if you work like this, this helps probably.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with elan, composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments.
So much about the short read sequencing technologies, and now we go further to the long read sequencing and starting actually with PacBio here. So PacBio applies a smart single molecule real-time sequencing approach. I think the keywords here, it's extremely sensitive, so it was developed and main application is for medical research actually. And it is also able to detect methylation by different fluorescence pulses. The principle here is, as you can see, that a polymerase, this is the green molecule here replicates one DNA molecule. So this is why this method is so sensitive. And it uses to detect the fluorescence a zero mode waveguide, which is basically a glass capillary that's small enough to guide the fluorescence to the detector. This is all embedded in another microplate of flow cell, which has a lot of very small microwells. And in PacBio we have now two sequencing chemistries that can be applied. The one is the continuous long read sequences, or CLR, which is the first and the older one, where you can use really long inserts up to 175 kb. The principle is that you attach this bell-shaped adapter to this insert, and then you start sequencing this. With the CLR, the problem is that the accuracy is only 90%, and you have multiple molecules but only single reads, so the coverage of the same molecule with CLR is rather low. And the preparation of DNA or RNA has to be really good, and demands a bit more work and caution than Illumina. So Illumina is very forgiving with bad RNA quality, for example. PacBio, Nanopore are not. The newer chemistry now is the CCS principle, the circular consensus sequencing, where you also use these bell-shaped adapters, but in the end you have smaller inserts, and you have this circle that builds up, and you can do several rounds or sub-reads. So this is, if you do hi-fi read sequencing, then you always extract those sub-reads. And there you have a single molecule but with multiple reads. So this is highly accurate, and you don't need Illumina sequences there. So the quality is to compare. So regarding RNA, this technology is a bit limited regarding the quantity and the quality of RNA you need for that. Working with small individuals, well, difficult. To understand that a bit more, how that works, also to see the machines. Now PacBio works with Sequel 2 and I think a third chemistry kit, but I might be wrong there. I also included here this video that shows that a bit more in motion picture style.
Introducing the PacBio Sequel 2 system, the most advanced sequencing instrument for generating highly accurate long reads. This platform makes long-read sequencing affordable for all scientists by generating more high-quality data in a single run. Using the Sequel 2 system, scientists can be confident in their discoveries, made possible with comprehensive views of genomes, transcriptomes, and epigenomes. The Sequel 2 system is powered by single-molecule, real-time, or smart sequencing technology. Smart sequencing uses the natural process of DNA replication with these key advantages. Long reads allow you to readily assemble complete genomes and sequence full-length transcripts. High accuracy provides over 99.99% accurate sequencing results for data you can trust. Uniform coverage enables sequencing through regions inaccessible to other technologies. Single molecule resolution lets you capture sequence data with over 99% single molecule accuracy. And epigenetics can be explored through direct detection of base modifications during sequencing. Here's how smart sequencing works. First, from any sample type, ranging from viruses to vertebrates, DNA or RNA is isolated. Next, a SmartBell library is created by ligating adapters to double-stranded DNA, creating a circular template. Primer and polymerase are added to the library that is placed on the instrument for sequencing. At the core of smart sequencing is the SMART cell, which contains millions of tiny wells called zero-mode waveguides, or ZMWs. A single molecule of DNA is immobilized in the ZMWs, and as the polymerase incorporates labeled nucleotides, light is emitted. With this approach, nucleotide incorporation is measured in real time. With the Sequel II system, you can optimize your results with two sequencing modes. Use circular consensus sequencing mode to produce highly accurate long reads, known as hi-fi reads. Or use the continuous long read sequencing mode to generate the longest possible reads. Smart sequencing has versatility across a broad range of applications, including whole genome sequencing, variant detection, RNA sequencing, to resolve complex populations, targeted sequencing, and epigenetics. From small labs to large initiatives, scientists around the world are using smart sequencing to generate data they can trust. Their findings are yielding new biological insights to improve understanding of human health and disease, propel agricultural advancements, support conservation efforts, and combat infectious disease.
All right, I hope that is a bit more clear with this video, and now we can switch to the technology I like most, which is Oxford Nanopore sequencing, also because it's a bit like Star Trek, the future is now. So you probably know that Oxford Nanopore Technologies applies nanopore sequencing. So you have this biotechnology manufactured pores that sit in a membrane, and then basically the changing ion current is measured that allows to detect the incorporation of bases. And well, I think ONT is really not to beat is in the size on application of the devices. So really mobile, and this is one of the major advantages there. I also included here a video because to understand this a bit more, this is the best way.
Oxford Nanopore Technologies has developed nanopore-based DNA and RNA sequencing technology, designed to provide on-demand biological information to any person in any environment. Protein nanopores are tiny holes that in nature form gateways across membranes. In our technology, protein nanopores are embedded into a synthetic membrane, bathed in an electrophysiological solution, and an ionic current is passed through the nanopores. As molecules such as DNA or RNA move through the nanopores, they cause disruption in the current. This signal can be analyzed in real time to determine the sequence of bases in the strands of DNA or RNA passing through the pore. During sequencing, the nanopore analyzes the entire fragment of DNA or RNA that's presented to it, so the read length is directly related to the length of the DNA or RNA in the sample that has been prepared. Users can influence their read lengths by choosing the right preparation methods for their desired experimental results. Standard extraction methods readily achieve reads from the tens to hundreds of kilobases, and gentle extraction methods devised by community users have achieved read length in excess of 2 megabases. Long reads provide a more unambiguous approach to mapping a DNA or RNA sequence, enabling much simpler assembly. Using nanopore technology, you can sequence DNA and RNA directly, rather than through a copy or synthetic strand, without the need for a surrogate marker. This means modification information remains intact and is included in the signal information provided to the user during the run. As PCR isn't necessary for nanopore sequencing, amplification bias is removed and library preparation workflows are simpler.
So basically one of the major key features here is direct RNA sequencing, which is of course very interesting, linked to the small size of the devices. So who knows, that could be you soon on our planet directly sequencing RNA from different species. The system with the MinION device here was already on the space station in action. The SmidgeION here I think is now operational, and that makes it really feasible to use that system to go into the field and sequence DNA or RNA remotely. And if you can guarantee sterile and clean RNA extraction, then this is really feasible. So this is a work from a colleague of mine, Stefan Prost, who developed actually this field chamber also for library preparation, etc. But I'm really curious how the direct RNA sequencing works, which I definitely plan to do. Working with the MinION or with ONT can be a bit difficult regarding the species, so I made the experience that using it for different groups of insects can have quite some different, let's say, achievements in sequencing output. So it's really important there to actually test in advance the specimens you plan to work on. So if a colleague gains super long reads, 20 kb, etc., from caddisflies, that doesn't mean that you can achieve that with hymenopterans or with robberflies, for example. So the system, for me, one of the disadvantages is really that you need to test that, and that a lot of work needs to be done by basically the user figuring out what's the best way to apply the system. But direct RNA sequencing, that's a cool part. We need more direct RNA sequencing in Venomics, that's for sure. How much material I do need for the RNA extraction now to prep the lib, you ask? Well, let's talk a bit about those numbers. We compare those methods a bit more detailed now. For the RNA extraction, I can say that my experience is that you really have to test different systems. So you can use column-based kits, you can use trisol-based methods, adapted or not. With trisol, of course, you can separate very nicely the RNA, DNA, and protein fractions. So in the end, I relied mostly on classic trisol RNA extraction and could also work with rather low amounts. Actually, also students of mine used this method quite neatly. So in the end, you have really to test what works best for you and what is maybe also better established in your lab. For the long reads, as said, you really need high quality RNA. So this demands really a very thorough treatment of the specimens. You need to ensure cooling chain. So for long read or direct RNA sequencing, the whole process is not very forgiving for errors that could, by any chance, degrade your RNA or impact the quality. So maybe not really something for expeditions. If we talk about total input quantities, then Illumina is, I think, the way to go if you have not much material. That's also one reason why I primarily used Illumina so far. So the average input is 100 nanograms, but there are ultra-low protocols where you can go down to 10 picograms or 10 nanograms. While for PacBio, I checked those numbers actually, 300 nanograms, and for nanopore it's 500 nanograms. So in general, I made also the experience that with nanopore you need more material than for PacBio. Important is here also to check the quality of the DNA. So for that is the RNA integrity number, the marker to check, which should be above 7, which is optimal. Basically, you measure the ratio of 18S and 28S ribosomal RNA to see about the extraction quality and the RNA integrity. But I used also samples lower because I didn't have any chance to obtain, for example, more material or new material. So it depends a bit on how you treat the analysis and how you interpret your results if you have maybe lower quality RNA as input. In general, of course, shitty input in, shitty output. That's also unfortunately a rule. maybe about the written numbers. If you check your RNA quality, you normally obtain like from tape station or chip-based quality numbers, and also those peak pictures. Important is here you have 18 and 28S peaks. This is spider, the wasp spider Argiope. You sometimes don't have those for insects. Yeah? So there you have only an 18S peak, and most sequencing companies then tell you, oh, don't use that, the RNA is no good. And then you can say, yeah, it is. So some insects group have this weird behavior that the 28S RNA has kind of secondary structure with the sequence part in the middle where it often breaks. And then you have only an 18S peak because the halved 28S also goes into this peak. So keep that in mind, that this can happen. And the sequencing itself is then outsourced in most cases. Always check best deals. Sometimes your institution has a good deal, or you can join from another institution the deals. The sequencing depth always depends a bit on your experiments. So if you do have a differential gene expression and you really want to go into expression levels, then I would rather aim towards 50 million reads. If you want to do a general venom composition description, depending on the grant you have, you can start already with 25 or 30 million reads. That depends really on the question you have. I would always intend to do more than needed, but keep in mind that if you do direct sequencing here or long reads for the quantification, you might need additional Illumina reads, because so far the quantification of long read RNA-seq is developing, because you simply have less molecules, longer molecules with less. The expression level statistics is still in development. Assembly. We talk now a bit about the read assembly, and I should say in the beginning that this is a topic that would demand actually an own lecture to cover that appropriately, so forgive me if I simplify here. In general, if we look at the different platforms, we can say that the first step is very similar. So if the reads come from the machine, you do a base calling and a quality filtering. That means basically those reads with bad quality readouts from the signals are filtered. Then you have to do that also normally if you start pre-processing the reads yourself, just to make sure. What we do have to do is the adapter and primer trimming. So for Illumina short reads and for the long reads using CCS and ONT, we have to trim the adapters. For the direct RNA sequencing, we have to trim the primers, so they're just primers added. And then we should do a contamination check, which is more or less difficult, especially if you work on a de novo species. But if you have, for example, a bulk of species you send to a sequencing facility, it can well be that there are sequences in one lane or one flow cell, and then inter-contamination can occur. You always should double check there, normally using simple blast to check if you find 100% identical matches. Hopefully you don't, because otherwise you have to contact your sequencing facility. And then we have also a read error correction, especially for the longer read sequences like PacBio CLR, where the quality is not that high. You would polish basically with Illumina reads, for example. For the long read and direct sequencing, the working flow is a bit younger. So the software for this is not that extensively existent in different versions. For the Illumina analysis, you really have a hard choice because there are so many possibilities. So first comes the base calling, and then the quality control, quality trimming, and then you would go into the read error correction, as I said. I don't go too much into detail here because actually we would have to do a hands-on session looking at the data. So this part or this lecture part on assembly is maybe or appears a bit superficial to those who did that already. Just as a side comment. For the Illumina short reads, we have actually plenty of publications that compare different quality and filtering software or tools. I gave you here the most recent papers and also most recent quality and adapter trimming tools. I think most established is Genomatic, which is included also in Trinity, the most commonly used assembler, at least in Venomics, I think. And here you have also an overview of all those different software tools you can use to do that with Illumina. In general, I think there might be differences depending on which tool you use, but I think more importantly is that you know what to do and that it kind of implements in your pipeline what you use. So if you use Genomatic or another tool like Condetri, which basically do the same, then that's okay to use one that's maybe not that established. It really depends on how your own working flow is set up. Let's talk a bit about the assembly. For short reads, for Illumina based, normally debrugging graph based assembly procedures are used and tools that implement those algorithms. So basically what we do is that we have all these millions of reads from the machine, and then we break them down into smaller reads, so-called k-mers, and you have a k-mer of 7 here. We look at that. No, it's 6. So 6 nucleotide is a fragment is a 6 k-mer. And what you then do is that you basically take those k-mers and try to arrange them in a way that you end up in more contiguous sequences. That sounds quite simple, and you can see here that's, yeah, it's quite simple here. But then you soon have different paths depending on which k-mer you add. So you can see here you end up in different paths which end or result in a different consensus sequence in the end. So basically what the debrugging graph algorithm does is to use graph-based mathematics to find the most optimal solution, to go through these k-mer possibilities, always arranges the k-mers plus 1, and then you end up in your transcripts. This is actually not that easy. It's an NP-hard problem, so there is not the most optimal solution to find. So it can be that assemblies look different depending on what you use. So basically keep in mind, if you have an assembly, that this is not an optimal solution, it's an approximation. And important is also that sometimes debrugging graph methods are combined with consensus overlap method. That means longer sequences are taken to break that bit up, and then you can rearrange a bit. And you find 2 papers describing that here below, where they describe that a bit more in detail. I don't know how much you know about the Euler paths. So this is basically finding the best way in too many possible solutions that you could end up. The evaluation of assemblies is therefore really important. We really have to keep in mind that assembly is not error-free. So the data quality of Illumina, so that the read errors coming from the machine are rather low. So the quality is really good. But the assembly is the part where it really starts to become a bit more problematic, where we need to be aware what can happen. What can happen are, for example, chimeric transcripts. If you have particular in Venomics multigene protein families where you have similar variants and you break that up in k-mers, you can imagine that there are kind of chimeric sequences that are basically virtual but don't exist in reality. Some assemblers are aware of that problem a bit more, like different isoforms. Some perform better, some perform less. But important is to keep in mind that this can happen and that you also check for that. So one trend that is to foresee is that actually multiple assemblers are combined in pipelines, so you end up in a multiple assembly from different assembler tools where the assemblies are merged and redundancy is taken out. This is also based on the fact that there are a lot of publications showing that assemblers really perform differently depending on what data set you assemble for DNA and for RNA based input. So we used in the recent time the Oyster River protocol, which is a multiple assembler, using also the k-mer approach for de novo transcriptome assembly. And in this assembly software, there are three assemblers combined, which is Trinity, Transabus, and then Spades. So maybe try that out. This is also available via Docker image, so this is more easy to install, at least for someone who knows a bit how to use Docker. And this is now basically the workflow that we normally use. We have the pre-processing, then the de novo assembly, and then the evaluation. And this is actually a paper where the colleagues compared the performance of different assemblers. So the take-home message here is, if you rely on one assembler, that might be fine, but be aware that there are limitations. There may be overestimation of isotics, so different contexts, some overestimated. You have to check that a bit. And here comes now the important part. If you have the assembly done, you did all this working flow perfectly neat and were very careful, conservative, exclude all the bullshit that might be bullshit, and then you have the pre-processing done, the de novo assembly done, and then you evaluated also that there are no chimeric transcripts and that the data looks all good. So you have basically the transcripts, and then you normally use that in Venomics to match that against a specific database against the mass spec results. So you basically identify the smaller fragments coming from the mass spec against this database. And here it's important to be aware that also, if you did a good job, it can well be that you might have artifacts of the transcripts. So basically transcripts that are not real in your de novo transcriptome, which can create false positives. This is especially the case in Venom proteins that looks so similar and come from multigene families. Or if you think about Norteins, the family that has those cysteine scaffolds, they occur quite commonly in the genome. There are a lot of similar sequences and proteins that you might cover, and if you have such a diverse family, then misassembly might be overlooked, and then you have to be really careful linking the proteome or the transcriptome data to the proteomics, actually. For the long read platforms, it's normally an overlap consensus technique that dominates, but linked to a k-mer approach where well, I can say a bit more sloppy, larger k-mers are chosen. And then you can assemble that, and very often if you use, for example, CLR, you have actually to polish with Illumina reads. This is the case also for nanopore sequencing, that you often have to add Illumina reads actually. The advantage of the long read platforms is, as said, that we have full transcript lengths. So this actually takes out a lot of possible errors, particularly on chimeric artifacts. So you avoid the assembly artifacts because you don't break up at k-mers. You use basically long sequences, and then you rather do consensus overlap, which equals pretty much aligning those longer sequences. And the cool part is that isoforms are directly sequenced. They're not predicted from the assembly. They're not gene models. They are basically the isoforms you find in that tissue. The disadvantage here is the sensitivity and sequencing quality that is a bit limited. And of course, as already said, that you need a lot of input material, which is, at least for my organisms, the limiting factor. Right, so this graphic here summarizes that a bit, maybe from a different graphical perspective now. So this is copied from this very nice review on RNA sequencing, the teenage years, where you can see here where we have the genome, and then we have in gray this transcript X and in red the transcript Y. And you can see here now the case for the short reads, where we see reads that actually map to both red and gray, where we have ambiguous read mapping to the exon. So there would be in an assembly in artificial transcript constructed chimeric transcript, while these here are kind of assembled unambiguously. The same can also happen with the isoforms. There's ambiguously the reconstruction of the isoforms, and the only chance to identify that actually is either if you have Illumina reads, you have the genome, or you use basically the RNA-based long read sequencing technologies, because there you can see you don't have those artifacts from the short read sequencing. So this is very important. And last slide summarizes this whole process again. So you have the tissue of interest, then you do the RNA extraction and amplification and sequencing. If you use direct RNA sequencing, you just ligate the adapters, and you have the sequencing machine. You end up in paired end reads for Illumina and the longer reads for the PacBio and ONT techniques. And then you have all the small reads, and either you assemble them de novo, as we discussed, or if you have the genome, then you would map them to the genome, and then you can identify the transcripts. If you have the long read sequences, then of course you have already the transcripts. I can also emphasize this publication here, the RNA sequencing data, Hitchhiker's Guide to Expression Analysis. It's a bit focused on Illumina sequences because it's a tad older. But yeah, so basically this was assembly in a nutshell, with the pro and cons, a bit focused on RNA. And with that, we go towards the transcriptome annotation. So the transcriptome annotation is basically that step where you try to establish what the transcript is, basically a gene model relying on also genomic data. So the first step is normally that you predict the coding domain sequence for the transcript, which you have of course many depending on the transcript length and what it is, where you translate from the nucleotide to the amino acid level. And again, there are many tools that can do that. I think the most commonly one used is Transdecoder. Transdecoder is based on the length of the open reading frame and the nucleotide composition, and tries to optimize towards this. The nucleotide composition is estimated. And very important here, Transdecoder can actually do a bit more. You can also use data from actual databases such as InterProScan and BLAST to add prediction based on real sequences. So this helps a lot. InterProScan has, for example, all the domain information from known sequences in the database. So you can increase the precision of the prediction from Transdecoder. And then the second step is the annotation. It's alcohol-free beer, where we try to do homology transfer from the amino acid sequences. We can also use gene ontology terms and also the feature annotation, for example, domain features. So what we can additionally do is that we use more tailored database searches for that. For example, you can do own hammer profiles of proteins you're interested in if you have a minimum number of sequences. Keep in mind, hammer only is working if you have enough sequences in the core profile to train and to search for other sequences. Or you can do blast search against, for example, known toxin databases. So what I normally do and combine, I normally do that step actually manually. So I manually search against InterProScan, but I also search via blast against UniProt, ToxProt, and then I have customized databases for the Venom proteins I'm interested in. And then you end up with the annotation for the sequences depending on the matches. Important here is many annotations are false or misleading. So if you end up with a blast search, for example, and then you have transferred that into a table, and then you have this column annotation, you end up with a lot of annotations that are actually not correct. So do not use blast or hammer search results blindly. Exclamation mark. Really go into the sequence, and yes, I'm aware this is manual work, but this is actually important. So you have to check what the result is and how the sequence looks like. If you check that, you will see that a lot of false positives are given out, which are actually something completely different. You see that normally if you combine the annotation results from different databases. So if you use UniProt, ToxProt, and maybe a third one, maybe more species specific, and you compare that, then you see, okay, this is actually nonsense. Especially ToxProt gives a lot of false positives because it only searches against toxin candidates. A side remark here is be aware that the e-value is dependent on the database size. Rather use independent scores, independent from the database, such as bit score, which is linked to the alignment of your string that you search against another sequence. So do that, it's really important. And also important is align and check the matching sequences. So if you have a manuscript where you discuss, for example, candidates for phospholipases A2 or hyaluronidases that you find in your data, actually take those sequences, add other known sequences and align them. You can see that still, because maybe one part of the sequence is similar, that matches your blast or your hammer, the sequence is actually also false positive. And what you can also do is to compare results from a specific toxin database against a normal database, including also non-venom proteins, which allows you in difficult cases to identify maybe false positives if you see that actually the match against the normal housekeeping gene is much, much higher than the match against tox prot. So that's what I normally do if I use body tissue and venom gland data or transcriptomes, then I always compare those. So the annotation is really that step where you cannot shortcut so much and you really have to dig in manually a bit to be sure what you talk about. That's how it is. Quantification and expression levels. As you can see, it's rather a brief summary. This chapter could easily take a whole lecture itself, and it becomes very fast, very mathematical. So I'll try to break it down that you understand a bit about the principle in which quantification statistics is normally to use. So important is here, we work with a transcriptome, so we have RNASeq data. So we have assembled now the transcripts, and then we use those transcripts to map the reads against the transcripts. Yeah, normally you would do it with the genome if you have one, which is more precise in many ways. But with a transcriptome we do it like this, and then we basically end up in a read count table in which all the data are parsed. You don't do that yourself. The software does it, which is the read mapper. So in this case we have 3 transcripts, and it's not 100% precise, but let's say transcript X is twice the size than compared to a transcript Y and Z. Then we have 8 reads mapping to transcript X, 4 each to transcript Y and Z. And then we normalize for the length, which is important. Here we have a ratio of 2 to 1, because if you would treat them like equally, then of course more reads map to transcript X, but this is also the reason because it's longer, so longer transcripts, more reads mapping. And then we end up with a normalized length here. And then we normalize for the depth of the sequencing library, so the total number of reads that map. Let's say for computational reasons here 1000 reads map in total, then we have normalized read maps here of 0.4 for transcript Y and Z and for transcript X. Important here is that only if reads are mapped to the genome, you talk about gene expression. If we talk about transcriptome expression levels, then of course it's also the transcripts and not the genes that are higher expressed. Check that after you know that now. Many people use actually the wrong terms there. Important here is that we have different statistics to calculate the expression, and actually that topic caught me at one point so much, and it was so much confusing things to read that I wrapped that a bit up in manuscripts that you see here. So generally I would advise you to use TPM. You still see a lot of FPKM calculations. The major difference is that TPM normalizes short but highly expressed transcripts more realistically and covers them better than FPKM. This is reasoned by the way how you calculate those values. Going more into detail would be too much, you know. And TPMs of transcripts sum up to same proportions within samples. That means you have in the end similar numbers, but keep in mind TPM and all of those here are not really designed to allow for comparison between different samples. That's really important. For that you would need a different gene expression analysis, which is something else. And to give you an example how you can do that, I would start with the maybe bad example from a manuscript, older manuscript of mine, where we presented the expression of venom gland transcriptome on the body tissue transcriptome for remipede crustaceans, where we still used FPKM. That was 2017. And you see here that we show those results beneath each other. And this is a way I would no longer actually present the results for a transcriptome expression analysis, where you don't really do a real differential gene expression, because you cannot directly compare those numbers. And this is a bit intriguing to do. So we actually didn't compare them directly. So as you can see here, we numbered the most highly expressed transcripts, and then in relation to each other, we said, okay, chitinases are the most highly expressed family in the venom glands, and then here it's proteinases as one. That is okay if you do it like this. But in general, I would do it a different way now. I would actually put them in relation to each other. So an indirect proportional comparison, where you have here, for example, 3 venom gland samples from hymenopterans, 2 solitary bees and the honeybee, where you can directly compare now the composition because you give the relation to each other. And these are also TPM values. If you want to do a proper differential gene expression analysis, I mentioned already that you need to have sufficient replicates, which is important. And the replicate numbers are really crucial to end up with a significant and robust statistic. For that, different calculations are used. For example, trimmed mean of M values, TMMs, which where you take basically the medium expressed genes as a scaling factor, their log fold changes, and that allows you to compare between samples. So this between sample comparison is now doable because there are a certain relation used by medium expressed genes. There are many software packages that do that. I think some also rely on the general direct read counts. And you can see here in the workflow that the FASTQ file is basically the transcriptome data and the raw files. And then you have different mapping tools that basically you can use to map first the reads and then do the statistics. There are also kind of pseudo mappers now that use kind of pseudo alignment to directly estimate the transcript expression. These are, for example, Salmon or Callisto, which perform quite similar and equally to the true read mappers, which are much more computationally intensive. Just keep that in mind. And I hope that was helpful to understand some basic principles. And then, of course, it depends a lot on your experiment and what you want to do. And if you take home message that TPMs are the way to go if you handle one sample or more specific sample without a proper differential gene expression analysis, then this is already good. And it's even better if you realize that the differential gene expression analysis normally works on or with genome data. Otherwise, it's a differential transcript expression. All right, stay with me. We're almost done. Yeah, bring on the genome. So we talk now a bit about the genomics aspects. So basically about this here. And of course, one quite simple use of genomes now, you probably understand why it is good to do so, to include a genome, to do a genome-guided RNA-seq analysis, because it simply eliminates a lot of artifacts from the de novo transcriptome assembly, which of course, if you're interested in the venom composition and don't want to go too much into detail in the evolution of the venom proteins and the underlying genes, then this is fine. But we can use the genomes, of course, in a much more detailed way to understand better mechanisms that drive venom gene evolution. Just in general, also very superficial, we have different mechanisms that drive venom gene evolution. I think the two major ones are gene duplication and single gene co-option. And then finally horizontal gene transfer, which is a special case, of course. Let's start with the gene duplications. Here we have the DNA, which is the genome with the genes and their control regions. And we have now here body tissue in gray, which represents actually multiple different tissues from the body, not only one in the best case. And in yellow here, the venom gland. So we see here that we have two genes, one gene that duplicated, and now we have the original gene and duplication number 1 in the body tissue expressed. And now that changes because one of the control regions of duplicate number 1 is altered, and this gene is now expressed in the venom gland. So basically this gene and venom protein was not present in the gland before. So this is a recruitment event where probably also a new function is involved. And then we have a different case where we basically have two genes that are expressed in the body tissue and in the venom gland. And then we see, okay, there is evolution going on, and suddenly the control region for the body tissue variant is eliminated. This gene is actually lost in the body tissue, but remained in the venom gland and evolves further, maybe also with new functions. This is what we call a restriction process. And then we have the single genes, where you have a single gene, but you can have, of course, multiple control regions for the expression in different tissues. And in that case here, we have one gene in the venom gland expressed and in the body tissue. And then by duplication, for example, of the control region, we can see that this single gene is co-opted for the venom gland and higher expressed. But there's also the possibility that both genes are present in both tissues. This one gene, with both control regions, and then for the body tissue, the control region is lost, and then we have a restriction of this single copy gene, which means that's only to find in the venom gland. This would be, for example, the unique genes, which are extremely interesting. The horizontal gene transfer is a bit a special case. It's a lateral gene transfer, so that means a gene from a different organism, for example, fungi or bacteria, is incorporated into the genome of an eukaryote. And then suddenly that is recruited into the venom gland. And interesting, of course, is also alternative splicing, which is a process for which we need proteotranscriptome data. So there the genome data is, of course, limited to identify this. Keep in mind, I mentioned that already a couple of times now, that many venom proteins belong to large multi-gene families, which are highly similar. And to understand the evolution, we need all gene variants that are to find in the different tissues. That means the gene completeness based on the different tissues is extremely important. And the cool part is that for most genome publications where a new genome is sequenced, of course, those different tissues are added to annotate the genes better. So those new genomes we currently produce in all this genome consortia, we have really the possibility to include all possible gene variants. That means genomes and multiple tissue RNASeq are crucial to understand venom protein evolution. And this is now, guess who's back? The thought you were supposed to hold on. This is a challenge with transcriptome-only studies, because if you miss many gene variants that occur, then of course inferred trees or inferred hypothesis, deduced hypothesis on the evolution of those venom proteins is not very robust. To give you an example for hyaluronidases, let's say we're interested in the venom hyaluronidases of water shrews. To robustly infer that, we need to know which hyaluronidases occur in that lineage and closer related, but also more distant related lineages. This is now an example which is really good, because for the placentals we know the arrangement of this gene family very well. So we have 7 variants that you can see here. Variant 1 occurs only in this clade here, and then actually is lost, pseudogenized, and is missing in this clade. The question now is, what is the variant we find in the venom in the water shrew? To know that for sure, we need to include all this data and to reconstruct the tree. And this data is based on genomes, proteomes, and transcriptomes. So this is the ideal example. So keep in mind that most or many multi-gene families are actually not well studied, surprisingly. We currently try to do that for hymenopterans, and man, I can tell you this is a mess. Venom allergens are driving us crazy right now. So high venom gene diversity that you infer by prototranscriptomics alone can be really misleading. As mentioned already, we have hybrid transcript, we have transcript artifacts, there are post-translational modifications. So it is really crucial here to incorporate also maybe the genome data to infer on the first base actually very precisely the gene variants, and then also based on that trees for those genes, and then to infer the diversity. Phylogenetic trees can also be misleading if you don't include all variants. So one problem is that some toxins are very short. So here we see examples for melitine, which is a short toxin peptide and the major component of bee venom for honeybee. And you can see here, these are all variants from the honeybee, and then here are the variants from the solitary bee, which look already quite differently. So in our study on the acute venom protein evolution, we looked a bit more detailed into the melitine using now existing genome data, because if you look at that, I personally would presume there are multiple genes actually, or that's highly diverse as gene family. And we found out actually it isn't. So we find in the existing or at the moment present genomes only one gene. In Bombus we find actually duplication, but it is outside of Bombus, it's a single copy gene which occurs also only in bees centulato. That was a bit surprising for us as a major venom component, and so we currently infer a bit the reasons in a separate study. Maybe post-translation modifications are a reason, or other artifacts also based on transcriptome assemblies. So we check that right now. Melitine, by the way, is a 2-exon gene, yeah? So this is a simplified figure. And interesting is, melitine is super short. As you can see here, we talk about the domain region with like 25 amino acids, depending on which species, even shorter, with a signal peptide 2 exons. This is extremely difficult to trace actually in genomes, and that is a shortcoming for short toxins, where the current prediction and also tracing methods are a bit limited. So we are currently developing a new approach where we use microsynteny, Basically, this what you see here is a microcentenary arrangement. So you have flanking gene regions where the gene of melittin is located. And this is very robust, actually. And you can use those flanking genes to actually find and trace the location of melittin, and then to see if melittin is present, because if you use a blast procedure as, let's say, the algorithm that is using the least assumptions, then you're partly screwed because it's so short. You end up with inconclusive blast results. And the postdoc who was I was lucky to have in my Hymenopteron project was Ivan Kolodarov, who developed this approach quite a bit and applied that also for the Hymenopterons now. And this is extremely helpful also because the orthology prediction of those similar gene variants is really difficult because variants are missed. And additionally, we can now also use machine learning approaches. This is a preprint we just published, and this is currently still work in progress. But there I just wanted to show you that genomes are actually quite important to improve proteotranscriptomics, also adding additional information, of course. But the complexity of all three pillars is large, and we still need to work on that to come up with either solutions. Right now, this, for example, is manual work, which we try to automate now. And of course, to bring in a perspective, you can do in the end much more. If you have the genome of an organism and then sorted out the venom protein families, the gene families, how they evolve, then of course you can do a lot of other cool stuff, like you can use the genome and alter it, for example, to identify the function of the toxins, and maybe also spatiotemporal dynamics, how they are expressed at what time during the ontogeny in which area. So there's a lot of that kind of work done with cnidarians by Ivo Moran and his working group, for example. And we can also, of course, see the ontogenetic fate of venom genes and their products. For example, in Hymenopterons, or a better example are actually dipterans. In dipterans, we have larvae which are very often venomous. They have also gland system attached to their jaws, and there are reports that they paralyze prey. I mean, a larva is immobile, pretty much, so this makes sense. But the adults are pollinators. So what happens there with the toxin genes? Are they kind of expressed differently or just silenced? This is kind of cool stuff we can only do and understand with the available genome. So this is really, really exciting times. And I think about the applied parts for which genomes can be used, you will hear other talks. Yeah, with that, I am at the end. I had to change only one because of a catastrophe from our daughter. So that's good luck. I would like to thank you, of course, also all the students and colleagues who were involved in the research of which I presented results here. The former Venomics group, which I was lucky to coordinate until the end of 2021. And in the end, of course, also all colleagues in EUVEN, which is really a great habitat to foster and extend research on venomous species. Maybe if you're more interested, this is my homepage. I also have a Twitter and Instagram account. Twitter is purely professional, and in Instagram, which is new, I actually do a biodiversity blog, multilingual. So it's auf Deutsch, it's in English, et en français, très cool. Maybe check it out. I write there a bit more about venomous species, but also non-venomous species of aquatic and terrestrial habitats. So thank you very much. I hope this was the right mix between details and simplification. As said in the beginning, I would have preferred to do that live, which is now a bit difficult, but please use the possibility to ask questions in the Q&A session. So 5:15, after the 20s, after the talks, I'm available here and will host your questions and take everything. I make sure that I'm prepared, and yeah, looking forward to receive some feedback and discuss maybe more details linked also to your project and linked to species you work on. Thank you very much, and I say I see you latest on the 20s. Bye.